All right, welcome to our seminar. I guess we will, let me start by introducing our speaker, Yale Goldman. He did his BS in Northwestern University and then PhD, is that right, in Pennsylvania? And then finally postdoctoral fellow at University College London. So he's been in the UK, and he understands my strange British accent sometimes, which is a good thing. He's very well known for his work in molecular biophysics, particularly dynin and actin locomotion and movement of biomolecules. He's also very active in the optical tweezers community, and we meet every year for SPA conference where we talk about optical tweezers and using light to study single molecule interactions. He's a wonderful speaker, and hopefully he can tell you something very interesting that you might not know about.
All right. Yes. Okay. Yeah, welcome everybody. Thanks for coming. Thanks for the invitation, Darryl. Really wanted to see the area here in his lab, so I'm very, very happy to get to come here today. See you guys. Actually, I meant to set the pointer here. This is better. Yeah, you can see that. Okay. I'm going to talk about helicases, and I thought that was an appropriate topic because they have some relatively novel ways of approaching how these work that are applicable to lots of different biophysical areas. So that's the idea of this talk. So I'm going to talk about first this helicase called DDX3X, and I'll tell you something about it. There's 17 chromosome-encoded proteins that are encoded on the X and Y chromosomes. So there's kind of male and female versions of this. But the Y homologs of these proteins have been hugely understudied. And the X homologs of these proteins often lead to diseases. So there's strong healthcare implications of this kind of research. So here's DDX3X and its Y chromosome homolog, DDX3Y. Here's the domain structure of this protein. It's got these two domains, which are called Rec-A domains, Rec-A-like domains, and this whole family of proteins called DEAD-Box proteins, because they all have this sequence in the active site where they're ATPases, where the nucleotide binds. And they have N-terminal and C-terminal domains, and then these strings of disordered peptides, which are called intrinsically disordered peptides, and you'll see that's important in what I'm going to tell you about. What do these do? Helicases unravel double-stranded nucleic acids. So these are RNA helicases. DNA helicases, of course, are important in replication, for instance, and many cell processes. This is an RNA helicase. And so the ribosome, when it's synthesizing a protein, only has room for a single-stranded message to go through it. So any secondary structure in the leader strand of an RNA between where the ribosome subunit starts scanning for the start codon, any secondary structure there has to be unraveled. So that's one of the jobs of this protein DDX3X. Also, they go into stress granules. So these are membraneless organelles in cells that appear when cells are under stress. And also, these proteins are thought to surveil the cytoplasm for foreign RNAs that would come in from a virus. So they have a lot of different functions in RNA metabolism. DDX3X and DDX3Y sometimes complement each other in development, and sometimes they don't. That means suppose one is mutated so its function isn't operating properly, then the other allele that's encoded on the other chromosome can take over for it. So that's called complementing it. And so it doesn't matter really too much of these exact, but in some cases, the Y chromosome, the Y version of the helicase will take over if the X chromosome one is disordered, not being expressed, and in some processes it doesn't. And there's actually something called a DDX3X syndrome, which only the girls get because if the DDX3X protein in a male is disrupted, for instance, from a DNA mutation, then the Y chromosome version of it won't complement, and so those are generally embryonic lethal. So you only see this, it's an unusual situation where you only see it hardly ever in a male. All right, here's part of the family of these dead box proteins. Here's DDX3X and Y, and this is another one which I'm going to talk about later, which is involved in initiation of protein synthesis and also unraveling secondary structure in RNA, in the messenger RNA. So first we're going to start with the DDX3X proteins, and I'll tell you something about them. Now, my collaborator in Philadelphia, I moved from Philadelphia to Davis, California about 2 years ago. So I was there for a long, long time. Started in the previous century. Can you believe that? So, this colleague of mine, Kathy Liu, wanted to understand the difference between the X and Y homologs of this protein. And it was known that under stress, these proteins help to form or go into so-called membrane-less organelles called stress granules, which you can measure by fluorescence and see these are droplets of protein and nucleic acids that don't have a membrane around them. And the X and Y homologs are very different in their behavior in this. You see here the Y homolog forms these much larger stress granules than the X version. So there's a difference, and we were curious about what parts of the code made such a big difference in their behavior. So there's a lot in here, which I'm not going to go through at all. I'll just tell you about one single molecule experiment that we did where we could see one of the differences. So this is a turf microscope, and I'm not going to go into that in much detail. It's a fairly standard thing nowadays. Total internal reflection microscope where you can see single fluorescent molecules fairly readily. And we have this kind of RNA sequence where it's a double-stranded here. It's attached by a biotin neutravenin linkage to the cover slip, and there's a single-stranded overhang. This is a typical target for an RNA helicase because it's scanning along the single-stranded, and then it's got to unravel the duplex part of the RNA. And now, if you just have RNA alone, we get these signals. So this is the donor fluorescence, and this is the fluorescence of the acceptor when we excite it with a red laser. Psi3, excite it with the green laser. Alexa 647, excite it with the red laser and look at the fluorescence. And this is the fluorescence energy transfer, which, again, I'm not going to go into because I think most of you nod your heads. You know what fluorescence energy transfer is. Okay, good. It's a microscopic ruler. And so you can see the fluorescence energy transfer, it's also called sensitized emission, is very low. And if we take the FRET efficiency with the RNA, it's only about 0.1. So it's the distance between the two is upwards of 7 or 8 nanometers. Now, as soon as we put the protein on, so it's DDX3X, what we see is a much higher amount of fluorescence energy transfer, and that can be dynamic. And there's a photo bleaching here, which we use to document the fact that this is a single particle. And so here, if you look at the FRET ratios, if I go like this, we get this very low component and very high component. So we imagine that now, these are cartoons, it doesn't really look like this, but you can kind of think about it that there's a low FRET condition, and then when the protein is bound, the FRET is much higher. And then as the helicase activity takes place, this component of the low FRET, it's even lower than the RNA by itself. See, this is 0.1, this is really 0 here, is growing. So the helicase activity is taking place gradually. And then if we look, this is this corresponding experiment for the Y homolog, the DDX3Y, and you see you get less development of that low fret signal. So again, we're finding at the single molecule level just what was found that I showed you at a regular confocal microscope level, that the Y chromosome version is much less effective in its activity than the X version. And that's what we learned here, among other things. Okay, sure. It's okay with me. I think we'll run out of time, but that's okay.
I'm trying to better understand because it's a helicase, right? So I assume it will be stuck when there's a structure they're trying to open. So this design is kind of a single open strand. So why it will bind there for like, I saw it's like 200 seconds, even a few minutes since bound there instead of just sliding away.
Well, one thing is to know is that these helicases are not processive. DNA helicases will get on a double-stranded helicase and burn through the whole thing separating, and that's required for replication, for instance, of the bacterial chromosome, the bacterial DNA. Here, the helicases have one chance to separate the strands, and then it dissociates. And so if you have a long enough duplex, it would take many attempts at separating the strands to get them completely separate. So what we're imagining here is this is not a complete helicase, but it's just separating the strands partially here, and that's why you get this low fret signal. So does that answer your question? It's a gradual process, and they're pretty slow too, but we're not measuring the complete helicase activity. It's just the production of this low structure that has a much lower distance, larger distance between the two fluorophores. I'm going to show you a lot more of this kind of thing measured a different way, so maybe it'll become more clear. All right, so here's what's thought to be the enzymatic cycle of this kind of helicase. So these are apo states. There's no nucleotide bound here. Two of them will bind to an RNA strand, and then ATP binds and it separates the two strands into single-stranded segments on the duplex. The adenosine triphosphate is split. That's the energy source for this. And the inorganic phosphate and the single-stranded RNA are released. And then you have the protein with a product, adenosine diphosphate bound. That'll dissociate, and then we start the cycle again. So we're interested in understanding this process in a much more detail, and especially the difference between the X and the Y homologs, as I said. So we found that using a different way of approaching this is much more facile, and we can get data much more rapidly on a commercial spectrometer that we didn't build. It comes from a German company in Berlin called PicoQuant, and I understand there is some PicoQuant instrumentation here in Irvine. So it's a confocal microscope, and in this case, it's a fixed position. The samples are not ligated to the surface, but they're free to diffuse in solution, and they'll diffuse in and out of this detection volume. And then we have a very brief laser pulse, about 150 picoseconds of 532 nanometer light, green light, to excite the Psi3 molecules. And then fairly shortly after that, so 50 nanoseconds later, there's a brief pulse of 637 red light that excites the Alexa 647. So we're separately exciting the green and red fluorophores on this sample. And then this is time correlated single photon counting. So there's photon detectors and measuring at the nanosecond, even below nanosecond level, so we can look at the lifetime of the fluorescence. That's what this decay is shown here. And so then we get these three streams of photons, which are, this is the donor fluorescence. So it's the fluorescence coming from the Psi3 when it's excited by the green laser. And this is the fluorescence that's emitted from the Alexa 647 under red laser light. And this is the sensitized emission, like I showed you before, that comes from fluorescence energy transfer. During donor excitation, we're looking in the acceptor channel because there's energy transfer. And each of these little spikes is a burst of fluorescence fluorescent emission photons that's coming from one molecule diffusing through the beam. So we keep the total concentration of the fluorescent substrates down in the nanomolar range or subnanomolar, so that on average there's one or less than one molecule in the beam at a time, and we see these single bursts. And from that we can get a lot of information, which I'm going to tell you about. So here, this is again a similar way of describing it. So we call this pulse interleave excitation. That was invented by Don Lamb, that term at least, and also Shimon Weiss at UCLA has done a lot in this. And another term for it is called alternating laser excitation. Okay, so you see there's two lasers, and using different switching techniques, you get every other green, red, and occurring in that experiment I showed you is at 20 megahertz, every 50 nanoseconds, swapping back and forth between. Okay, so here's the three streams of photons that I was talking about. So if the donor and the acceptor are close enough to each other, we'll get the donor fluorescence and we'll get the sensitized emission, as I showed. And then alternatively, when the red laser pulse comes in, we'll get the acceptor emission. So that's what those three. Now imagine what happens if the distance between the two fluorophores changes so that the amount of fluorescence energy transfer is decreased. Well, we still know there's an acceptor there because we can directly excite the acceptor. And that's different from if the acceptor were to photobleach, or maybe the two dissociated if they're in a complex. And so we can distinguish distance changes from disappearance of the acceptor completely. And that's important, as I'll show you. So in this case, we don't get, if there's no acceptor there, of course, there's no directly excited acceptor photons. Okay, so here's the equation which you all know about for the FRET efficiency. So it's the sensitized emission. So it's the acceptor photons under donor excitation over the total photons. So that's the simplest way of describing the fluorescence energy transfer efficiency. All right, now we get another signal from this, which we call the stoichiometry. And that's the total photons under donor excitation, rather than just the sensitized ones, over the total photons in both cases for both acceptor and donor excitation. And that S variable is 0 for acceptor-only molecules, and it's 1 for donor-only molecules, and it's somewhere in between if we have donor and acceptor in each particle that are diffusing together. And so that's important. That's why we call it the stoichiometry. That'd be half if it's one of each of the two fluorophores. And now we can plot the FRET efficiency against this stoichiometry variable, and we get these different peaks. So this is the donor only. The S is 1, as it shows here. Down here is the acceptor only particles. And in the middle, at about 0.5, these are duplexes that have both fluorophores in them. And this has many advantages, which I'm going to show you a lot of data of this kind of two-dimensional histogram. But one thing is that if you have something you're labeling with two fluorophores, you don't have to have perfect stoichiometry on each molecule, because you can easily separate out in this plot and look at only the doubly labeled ones, which are the ones in the middle here. So just experimentally, it's a big advantage. This is a good sample, right? It's almost all doubly labeled. This is a histogram measuring in this direction, and there's some single-stranded molecules here, but most of it is doubly labeled. And the FRET here is very high, so the two fluorophores are very close to each other, which we can see on this axis. Okay, so here's the way the kind of experiment goes. Over a 2 or a 5 minute interval, the lifetime decays of the photons gradually build up during a sampling interval of a few minutes. And then these are fluorescence correlation spectroscopy spectra, which I'll talk about quite a bit more later. Students love this because the experiment itself is very easy. If your sample is a good sample, you just put 200 microliters or even 40 microliters in a well on the cell multi-well plate, and then push the button. So I always say any child can do this. The students don't like it too much when I say that, But then so you get data all the time, and then the analysis takes some thought. So it's not a trivial thing to get some results, but doing the experiment is great, and you get data every single time almost. All right, so what do we learn? So as I told you, we can measure the number of donor-only, acceptor-only, and duly labeled ones. Oh, I was going to describe the internal parts of the detector. Now, is this all sort of super familiar because, or is this sort of some interest to this group, how it works? Yes, sure.
You need the lifetime to be more accurate about the threat efficiency to determine that, because you can do it with intensity.
Right. Yeah. The software that comes with this will do it either way. And as you know, there are many corrections to intensity measured fluorescence energy transfer, but those can be corrected. There's 3 or 4 correction factors, which are easy to determine, and then you get a quantitative FRET measurement, or you can do it from the lifetime, and those will match each other. So just as what you're suggesting, that both ways of doing it are facilitated by this, and they give the same answer if you do the corrections properly.
Do you need lifetime? Could you do this experiment with just alternating excitation, gated detection, and not determining lifetime, just having 2 gates?
Yes. What I've told you so far for FRET and for stoichiometry doesn't need the pulse laser, but I'm just going to go into something which is a nanosecond fluorescence anisotropy decay, where you do need the higher time resolution. And then as a check, which is, I think, just what you're suggesting, as a check on getting the correction values for intensity-based FRET by using the lifetime, that's very valuable too. All right, so far this is what I've told you. Here's the 3 streams and the lifetimes, okay? So here's how we can measure the helicase, because we can count the number of bursts that come through that are doubly labeled versus the acceptor only and the donor only. And then when we add the ATPs, nothing happens, but when we add the ATP, you see the number of bursts, fluorescence bursts that come through that are doubly labeled decreases. So we can directly measure the true separation of the 2 strands. And donor only and acceptor only are particles that only have one or the other fluorophore in them. So that's useful to measure the activity. Here's the fluorescence correlation spectroscopy, and this is fluorescence cross-correlation spectroscopy. FCS, it has somewhat of a pejorative nature to it from the old days of Elliott Elson and Watt Webb, who invented this, where it was not at the single molecule regime, and the concentrations were much higher, and there was a lot of fitting of, and somewhat subjectivity to fitting with various artifacts like triplet states and double pulsing in the detector and so on. Here we don't have any of that, because if we cross-correlate the 2 red and green fluorophores, the triplet state is not correlated between the 2 fluorophores, and double pulsing doesn't count. So you see this really clean single decay fluorescence correlation spectrum where we can measure the diffusion. That depends solely on the occupancy time of the fluorescent molecule in the beam. We know its volume. So once we get this half-time of the FCS curve, we have a very good quantitative measurement of the diffusion constant, and therefore the size of the molecule. And lastly, which is one of the answers to his question, let me just go through this really quickly. So we have a fiber coming in with different colored lasers. I'm telling you about red and green, but the instrument itself has several other colors. A dichroic here that projects the light into the microscope sample, comes back, and then there's a pinhole here, which makes it into a confocal microscope and restricts the axial dimensions of the detection volume. And this is a polarizing prism, and there's 4 photon detectors. So we're getting both parallel or vertical and horizontal photon emission. And then there's 2 dichroic mirrors, one here and one here, we separate out the donor and acceptor emission. So we're getting these between these 4 signals, and one of the advantages of that is that we can get both parallel and perpendicular. And it's big enough there. It looks like a small change here between the red, which is the parallel, and the blue, which is the perpendicular. But this is a log scale with many decades. So the parallel is about 3 times higher than the perpendicular at the beginning here. And if we take the anisotropy equation, which is basically subtract these two, we get this decay, can fit it to an exponential, and that decay tells us the rotational correlation time, or how fast the molecule is rotating, which again depends on the size and shape of the molecule. And also, it's more sensitive than fluorescence correlation spectroscopy is for small changes in the size of the molecule. So that's a big advantage of doing this. There are commercial instruments that use continuous lasers, much cheaper than this. And so for a lot of this, what I'm going to show you, it could be done, as you were saying, with the continuous laser, but we do get some particular advantages using the pulse lasers. Okay, so here's some experimental data. We start out with mostly, so my postdoc, Shweta, I'm going to show you her picture later, does a very good job of annealing the two single-stranded RNAs to each other. So it's almost all doubly labeled here, and very low fret, as I showed you from that turf experiment. And we add the protein, and then we get a much higher fret in the doubly labeled. And there's also a little bit of ATP-independent helicase activity, one cycle of ATP helicase activity, which we expect from that enzymatic cycle that I showed you before. So there's some strand separation. You get more donor here and more acceptor only here than we had before the protein was added. And then if we add ATP, the fret intensity of the doubly labeled goes down, and also the amount of donor and the amount of acceptor goes way up. So we're looking at the real complete separation of the strands here, because we're getting the growth of the donor and acceptor particles. And so we get some partial separation of the strands where the fluorophores will get, and again, this cartoon is how we imagine it to visualize this, but it probably doesn't really look like that inside. But then the fluorescence efficiency of the doubly labeled strands will go down, and then also there'll be some true helicase activity where we can measure where the two strands come apart. And again here, just as we found before, the Y homolog is much less effective at separating the strands than the X homolog. So that's probably why it doesn't complement each other, as I talked in the beginning. Okay, now here's the FCS. Now this is probably something much more familiar to you, but just take a minute on it. We have the pulses of emission from the two fluorophores separately. So this is a cross-correlation, fluorescence cross-correlation spectroscopy of the two colors. And then depending on how long they spend in the detection volume, if in the computer we shift one with respect to each other and more, depending on how broad this is, this is the time axis, that will determine how rapidly this decays as a function of the time of shift of the two signals. And so you get a decay until the shift in the computer of the two signals with respect to each other doesn't provide any overlap between them. There's no correlation. And so you get this decline. So that's the basic idea of fluorescence correlation spectroscopy. But here again, it's very, very clean because there's no triplet or double pulsing. We know the volume of this from the optics, and we can measure it. The diffusion coefficient by Stokes' law goes as the reciprocal of the radius if it's a compact globular particle. And that's shown here. And the volume goes as the cube of the radius, and that's going to come up importantly here. So here's some experimental data on the fluorescence correlation spectroscopy. Again, I emphasize how clean this is here. There's no doubt about single diffusion and no artifacts. So it takes on average, about 300 microseconds for this RNA to diffuse in and out of the beam. The occupancy time is about 300 microseconds, which is what's expected from this size molecule, 27 kilodaltons. So we get the diffusion constant. Okay, then we add the protein, and we get this huge shift in the FCS curve. Now, this was completely unexpected. I told her, you're doing something wrong. It can't be. You never see this, or hardly ever see this in an FCS spectrum. This is 5 or 10-fold shift of the diffusion constant. And remember, the diffusion constant goes as the reciprocal of the radius, and the volume goes as the cube of that. So a 5 or 10-fold change in the diffusion constant is a 500 or 1,000-fold change in the size of the particle. So how can that be when the protein binds? So what we have found is that the protein binds in clusters. And it's not just 1 or 2 proteins that binds to the RNA, but many, many dozens of proteins are present in these clusters that are diffusing at this much lower rate, a millisecond or a few milliseconds, rather than a couple hundred microseconds. This was a big surprise. And also, it takes very little protein to get the diffusion constant to come from, this is 80 in the units of a micron squared per second, down into 10 or so. So only 5 nanomolar protein is producing these large particles. So again, it was a very surprising finding, which was hard for us to believe at first. What we found is that, I'm not going to take too much time over this, is that these intrinsically disordered domains are important for this big shift. And you may know that the intrinsically disordered domains of producing stress granules are quite important for multivalent nonspecific interactions between associative proteins. So it shouldn't have been, it was not really unexpected once we believed in these clusters that the IDRs would be important for this. So we can make what we call delta-delta, which has both N and C termini IDRs removed. So it's just the Rec-A-like domains or the dead box proteins. And when we do the FCS experiment, what we see is a much smaller groups of proteins. This is only 2 or 4 proteins bound rather than dozens here. So the size of these particles depend very much on having these intrinsically disordered domains. And we can quantify that. So here's the diffusion constant of the RNA and a control protein. Here's the diffusion constant, much lower, of the native proteins. And the truncated ones are in between, as you can see directly from this. And then something we did in that paper, which I'm not going to take the time over, is we could swap the IDRs between the 2 proteins. Is it the N terminal or is it the C terminal one? So all that data is here. But one of the ends is important for cluster formation. The other end is important for the activity. So I won't take the time over that. But again, we found if you cut off the IDRs, the activity is very low. So it has to go into these clusters to get a good helicase activity. When you do that, the amount of residual RNA that you get after a period, so the activity is higher for the X version than it is for the Y homolog version. All right, and then because we were very suspicious about this, we wanted to look, what do these particles look like in an electron microscope? So I said, well, why don't we take a snapshot of those? And she said, well, we tried already, but we couldn't see anything because the person who does the cryo-EM, our collaborator, his name is Kenji Murakami, said, and of course he wanted to solve the atomic structure by cryo-EM. So he said, well, I can't do anything because there's all these clumps. So he had already had this result by the time we got that. These are individual particles. They're about 20 nanometers. Sometimes with this protein, they'll form little strings like this, you can see. The Y homolog forms bigger strings. And this makes us think these are not liquid like stress granules because these would, by surface tension, become spherical if they would combine. They're not doing that. And if we take the IDRs away, the particles are much smaller, as we found with the fluorescence correlation spectroscopy. So this supports what I showed you before, that there's these cluster formations. We don't like the word aggregates. That has a pejorative meaning. So clusters. And this has been found with some other different proteins. We weren't even aware of that. And also, this is another way of looking at it. This is something called mass photometry. Why isn't that going? It's a movie, but for some reason it doesn't want to play. Let's see if I do this and go back to here. Here we go. So is there a mass photometer here in BLI or on the campus? This thing is fantastic because, again, it's very easy to use for measuring the size of particles. It's invented by somebody by the name of Philip Kukura, who's in Oxford, and there's a little company. And this instrument is a commercial instrument. It's only about $200,000, so you can get it on a supplement to your grant. All the young people sitting there, write a supplement for your boss's grant, and you'll get one of these. But it's great. It's, again, very easy to use. The contrast, there's software that improves the contrast a lot. And that's what's one of the things that's special about this. The interference between a reflected wave and the scattered light is directly proportional to the mass of the particle. So that's why they call it mass photometry. We measured 8 megadaltons for this. The subunits of this are about 70 kilodaltons. And the RNA, as I said, is a 20-some kilodalton. So that's many, many proteins in these clusters. You can see each one of them land there. And sometimes it goes light, that's when it dissociates. And we can calibrate it with ribosome subunits. And if we use the truncated ones that don't have the IDRs, we just get a much smaller particle on the mass photometry. So all this just is a very strong correlation or support for what we found by FCS. And so the editors and referees were happy with that in that paper. All right, so, oh yeah, now, they're not aggregates of disordered protein. And one experiment that shows that there's helicase activity in the cluster. And here's an experiment that we copied from somebody else, Rohit Papu in Washington University in St. Louis. They thought of this experiment for clusters that they found with a different protein. We can make clusters by mixing double-stranded component here that has only one of the fluorophores. So these are green clusters. And then these are red clusters, which is the same RNA, but it only has the red fluorophore. Okay, and measure the cross-correlation, autocorrelation, cross-correlation function. And there's no mixing. This is the cross-correlogram, the amplitude of it. Okay, but when we add ATP, what happens is the strands get separated and some of them escape from the cluster and get recaptured. So then there's a gradual, very slow increase in the cross-correlation, which could only happen, it only happens when ATP is added, meaning that the helicase activity is active. So then we get clusters with mixed red and green, which is what this shows cross-correlation in the FCS experiment. So they're active inside clusters. We call them RPCs, RNA protein clusters, foster the helicase activity. Okay, now, that's all I'm going to say about DDX3. I'm going to get back at the end about what's the point of all this and why do these things form these clusters. Then I want to talk about this other protein, which is EIF4A. It's in the same family. It's got this sequence homology in the 2 RecA-like domains, also with that DEAD sequence called DEAD-Box in the active site, but it has much smaller disordered ends. So it's almost the first experiment we did when we came to Davis. Somebody by the name of Chris Fraser works on this as a mechanism of initiation of protein synthesis. He's an expert on this EIF4A. This one, according to our previous results, shouldn't form clusters, right? Because the intrinsically disordered domains are much smaller, as you can see here. Those are the DDX3, X, and Y. So now, initiation of protein synthesis is devilishly complicated. I can never remember how this works. I'm not going to describe this to you, but this little tiny bit here is the part of the initiation where the EIF4A is that little ball, and it has some accessory proteins, G and B and E. And what? The pointer. Oh, I'm sorry. Okay, right. How long have I been talking where you couldn't see the pointer?
Just one slide.
Sorry. All right. Yeah, here. So here's EI4A, the helicase, but it acts with these accessory proteins to make a oligomer called EIF4F, and there's another one, EIF4B. So I'm going to tell you something about these activity of these things when they're grouped and when they're not grouped. But the helicase activity, it's just this one segment. And the point of this is that an authentic eukaryotic message is capped at its end with a 7-methyl G, which is actually put on the wrong way around. It's really a strange structure, but it has to recognize that cap in order to activate the messenger RNA and for the rest of this cycle, which is protein synthesis, to take part. And that's the function of these, not only unwinding the secondary structure, which is a helicase activity, but activating the messenger RNA. Okay, so, and what do we find? So here's what I showed you before with DDX, 3X, and Y. As predicted, when we add EIF4A, it shifts, but there's only 2 or 4 proteins bound to each RNA. So it doesn't form these huge clusters. And that's what we thought would happen based on our results that cutting off the IDRs from the DDX3 proteins would prevent or make the clusters much smaller. So that was pretty satisfying that with a completely different protein, we could replicate this kind of idea that the IDRs matter for making these groups of proteins. Now, there's all these other factors that matter, and I don't want to take too much time over this. I'll just tell you a tiny bit about them. So this is the helicase. This is a so-called cap binding protein. And some of this is controversial, how it really works. But this protein will bind to the 7-methyl G cap on an authentic RNA. Here's the start codon, and there's a stop codon, and then there's a poly-A tail in mammalian messenger RNAs. And sometimes it will have secondary structures. So this is going to unravel secondary structure, or the ribosome will never get through and find the start codon. G is a scaffolding protein. So it's got all these different binding motifs, and it holds together this oligomer of these different subunits, which is the active particle. And then B, this is almost all disordered. Here's a cartoon of what it looks like. It only has very small amounts of folded protein. Most of it is just random strands or disordered. It has an RNA binding motif and some other specialized segments. Okay, so how do these things combine? So first of all, EIF4A has hardly any activity. Add the protein, nothing happens. Add the ATP, you start to get some helicase activity, but very, very low, and the fret doesn't go down. So this is very different from what I showed you with the other helicase that I talked about before. But if I start adding these factors, then it does work. You see how we don't get clusters when we add the protein like I showed you before, but when we add ATP, you start to get helicase activity and cluster formation. So here's the activity of the full-blown complex, A, E, G, and B. Put them all together, double-stranded helix target, and here's the activity, and you're starting to get clusters now. So with all of these factors, the activity and the formation of multi-protein clusters of these proteins takes place. And now she separated out each of the different functions of these by doing individual and then in various combinations. I don't want to take a lot of time over it, but what you can see is if you add G by itself to A and ATP, there's hardly any activity and no clusters. If you add B, that's the disordered one. There's a little bit of activity. This is the doubly labeled again, so the decrease of this is the separation of the 2 strands. And then you see the diffusion constant is starting to get slow again. So we're forming some larger groups of proteins when we add the disordered protein. B is that one that's largely disordered, intrinsically disordered, to A. It binds. And then, but when we add all of them, you get a much higher activity and larger clusters. So there's a relationship between the cluster size and the activity, which we can see by varying these different amounts of these proteins and in combination with which ones. There's a lot of work in this, as you can imagine, but she could get through it very quickly because the experiments themselves are so straightforward. Here's a titration of the concentration of EIF4B, 10 nanomolar, 25 nanomolar, up to 500. And you see the activity gradually increasing. I don't want to take too much time over this, but the main point is you don't start to get good cluster formation or activity. Well, you start to at quite low protein concentrations, again, as I showed before with the other helicase. So it doesn't take much protein to start slowing diffusion down by a lot and getting activity. 50 nanomolar, here's the pink one. It's pretty good. And now this disordered protein, our collaborators had already known that RNA binding motif of the EIF4B is important. And there's the disease mutants of this, which they studied, and they picked out one that's very good. So we can mutate one of the residues in EIF4B, phenylalanine 135 to alanine. And that has a big effect. It was already known before we did this on formation of droplets. This protein also goes into stress granules. So, but that requires, they require RNA. RNA is necessary to form these stress granules. And the binding motif of the RNA to the protein is important for that activity. So here's what happens if we use this mutant F139A. You see the activity is slower. And you can see here, this is the rate of the activity. So it's significant if it can't bind the RNA as well. And also the same thing applies to the cluster formation. There's the native one, doesn't form clusters. So again, there's this relationship between groups of these proteins on the RNA and getting a decent amount of strand separation. And again, the mass photometry of these shows the same thing. These are the clusters with the wild type EIF4B, and then if you have the mutant with weaker RNA binding, you see much smaller clusters. This is the contrast. Okay, so anticipating, so this is unpublished, anticipating a referee question that we always get, okay, what does this have to do with cells? You're measuring all this stuff with purified proteins. Is this meaningful? So that, okay, we got to do some, is this any of this going on in cells? So here's some FLIM images. In the same instrument, we can look at the diffusion constant and the lifetime of the fluorophores. So what you're looking at is fluorescence of EGFP labeled EIF4B that was transfected into HeLa cells. And actually there's leakage, even if it's a TET promoter, even if you don't put the tetracycline in, there's enough leakage because we only need very low concentrations to measure the fluorescence correlation spectroscopy of individual molecules diffusing through the beam. So here's what we got. GFP only. So they made a cellular construct that just has the EFG, and it's showing a diffusion constant of 22 micrometers squared per second. That's about 4 times slower than what GFP would do in vitro in a regular aqueous non-viscous buffer. So 22 is about 4 times slower than what GFP diffuses in an in vitro experiment. But the EIF4B diffuses much slower than that. So if you would put the 4B into a cell, the diffusion constant of that size protein is about 40 micrometers squared per second. But inside of a cell, the wild type is only 0.6. Well, if we divide that by 4, suppose the viscosity of a cell is slowing down diffusion, and crowding is simply the cause of slowing of the GFP. If that applies to the EIF4B, then 1/4 of this 40 micrometers squared per second should be 10. But actually we measure 0.6. So there's a big slowing of the EIF4B, much more than what is expected from just the crowding in the cell. And very satisfyingly, the mutant diffuses faster, which you can see here in the FCS, in the slow component of this. So this is supporting, and here's this diffusion constant of the EGFP in cells, and here's the wild type and mutant. So you see the mutant is diffusing faster because it's not going into the same objects that are slowing this down, which are clusters. And it's not only just the other helicase proteins, but there's probably other factors in those clusters. But this shows that even in cells, this kind of slowing of diffusion by cluster formation. And what was really satisfying is the prediction that the mutant that doesn't bind RNA as well also diffuses faster, meaning it either goes into fewer clusters or the clusters are smaller, just what we found in vitro. Okay, so now here's our fantasy about what the point of these is. And how am I doing for time? Not too bad. All right, I'm almost done, so that works. So as I showed you, the protein binds to the RNA and does a sort of partial strand separation. But this RNA duplex is about 20 bases long. One cycle of this can only separate about 10 bases. And if the protein dissociates before another protein binds to the other part of the duplex, what will happen is it'll reanneal, right? And so you could get a cycle like this where you're getting multiple attempts at pulling the strands apart, but it doesn't really work because there's a reannealing, which is very rapid when the protein dissociates. So you need to do better than this to get the strands really to come apart. And so suppose you have a bunch of proteins in the cluster and they're all randomly attacking very rapidly the RNA, and stochastically or randomly, two of them or more of them will go through their enzymatic cycle at once. And so that's when you could get true full helicase activity and the strands will really separate. So this is, we don't have a good experiment to show this directly, except that activity in the clusters and the relationship between the clusters and the and the strand separation suggests this very strongly. But maybe you guys can think of a good experiment to show this is really what's going on, and that you really do need multiple random attempts at helicase activity to get this apart. And then the cluster formation and the relationship between formation of these clusters and the activity is important because these RNA helicase molecules are not processive and they only can do one cycle at a time, so it needs multiple attempts. So you have your cluster, and then we get, it does its job, and we're happy, and they're happy. Okay, so here's what I talked about. I'm not going to go through all this because it's basically what I already told you. We get these stress granules. We love this picoquant approach. It's very effective. And then we discovered these protein RNA clusters, which we really were unexpected. And we think they're very important for activity of all these different proteins. Are they crucial for initiation? But at least we know that it goes on in cells. So there's something to it other than just our biophysics experiments in purified components. This is our larger group in Philadelphia. There's 4 different labs here. The people who participated in this particular experiment, this Amber Yanas was the student. Kathy, I mentioned before, collaborator in the biochemistry department. Another student, Michael. And Shweta, she came with me from Philadelphia. I'm going to show you her picture again in a minute. Shweta and Amber are the first author on these papers. So far, we have 3 on the DDX, and we haven't published the IF4A yet. And my collaborators in Davis, this is Chris Fraser, Masa, and Nancy are investigators in his department. They're experts in initiation and supply all the proteins for us when we're doing this spectroscopy work. I purposely kept a much smaller group in Davis. So here's Shweta. She came with me, and so did Arpan. They were postdocs with me in Philadelphia. And UC Davis has the same rule as in Philadelphia, that postdocs can only be postdocs for 5 years. You probably have, that's probably UC-wide, this rule. So they're now in staff positions called assistant project scientists, technician. These are 2 undergraduates. Bioengineering is fantastic in Davis. So I put out a call for bioengineering students for zero mode waveguides and a mixing device, rapid mixing device, microfluidics, get really great students. So I'm very happy. These 2 are graduating. I think one of them is applying here to bioengineering here, but she's sure to be accepted I'll tell you that because our record is fantastic, so I don't have to. Well, I'll write her a letter, of course, but I expect great things from both of this. One more undergraduate is not shown here. A lot of our funding comes from the National Institutes of General Medical Sciences. Now, I have a renewal pending that was supposed to be renewed. It was supposed to be evaluated by the study section on November 5th. Of course, that didn't happen because none of them were allowed to go to work during that period. The lockout is over, but the exec secretary, who's very nice and friendly, says we're waiting for instructions. So there's no schedule for even evaluating that. So that's troublesome, and I'm sure lots of people here in Irvine are under the same pressure. There's a very large NSF program called the Science and Technology Center, and the one at Penn is called the Center for Engineering Mechanobiology. This RNA work isn't related to that, but we also do a lot of work on molecular motors and cytoskeleton, so that's that part of it. We did have one from the National Institute of Standards and Technology for ribosome work. Collaborators, I mentioned Kathy, Mike Ostap on molecular motors, Barry Cooperman on ribosome work, nonsense mutations and read-through, pharmaceuticals for read-through nonsense mutations. Aarhus, Denmark, Marco Capitanio in Florence, Ram Dixit is Washington University. He's also a main person of this CEMB, and other people in Philadelphia who you may know. So thank you for your attention, and of course I'm happy to answer questions. Did I answer your other question? Oh, good.
Sorry that the concepts that you need the cluster to do like the slide.
Yeah, me too, but how can we prove it?
Yeah, so I'm wondering if you have, I mean, you have a 20 base pair, like, so if you have 10 base pair, 20 base pair, 30 base pair, any chance you can see a different size?
Yeah, we've done a tiny bit of that, but the approach you're talking about is very, very open-ended, because you can imagine not only the size, but how about the sequence? And the sequence in the single-stranded overhang, the sequence of the duplex. So what we know is that the helicase activity is much faster for 10 base pair duplex. So that fits with this idea. But you still get cluster formation even with that, but the helicase is much higher. So that does fit with the idea. The particular sequence that's the standard for EIF4A is different from a different lab than the one we were using for DDX, 3X, and Y. So we've tried the corresponding experiments on the other ones. It's the only thing we've done with different sequences, and you do get some different results. So it depends on the sequence, particularly in this single-stranded overhang. The one we used on the DDX is all poly-U, whereas the one that their favorite one that gives the highest activity of the initiation factor is a mixed basis. So that does matter, but in detail, I can't say more about it than that. But as you can imagine, it's very open-ended. And then Chris told me, in fact, yesterday, well, what happens in a cell lysate, right? You can grind up cells and get, it's active in translation, it's got all the factors there, but you can spin out the ribosomes and put your own ribosomes in, take the ATP away. So again, how many variables are there in that approach? And we think we need to have a specific hypothesis and question and sort of target that. Otherwise, it just mushrooms into something you can't make any sense out of because there's so many different possible experiments. Yeah.
So the cell experiment that I don't quite understand, because it looks like you've got lots of signals, at least there's quite a few of your target proteins in there swimming around. And they only cluster if they actually encounter RNA, right, in the cell. So but it seems from your cross-correlation experiments that we've had a very clear shift, and that's determined by the diffusion coefficient. So that would mean that the majority of those labeled proteins would be bound to RNA. That seems like funny, because it looks to me like the majority is talking unbound, just swimming around inside solid space, right?
Well, the only thing we actually observed is the RNA in that particular experiment. So there's a half nanomolar of RNA. Most of that goes into clusters, even at 5 or 10 nanomolar of the protein.
You inject the labeled RNA into the cell?
Oh, no, I'm sorry. What I was talking about was an in vitro experiment. In the cell, we're expressing, the only thing we've done here in cells is the EIF4B, the disordered one, which is, yeah, GFP. And with the DDX3, we had the same discussion with the referees over the other helicase. And so we did it before we submitted, but we had M-Cherry labeled DDX3X. And we saw the same thing, much slower diffusion than you'd expect just from the viscosity. So,
So does that mean that you think that most of those labeled proteins are bound to molecule? Because to me it seems like
there's a lot of it. It's most, you can see a faster diffusion. So we see slow diffusion like this, but there are faster components of the diffusion. So we can detect that. And I don't have a good handle on the proportions, but there's a substantial amount of it going into the clusters, whether it's most or not, I'm not sure.
Now, how long do you think you take one measure? You've got this beautiful cross-correlation curve. How long does that take per measurement to get?
So those are either 2.5 or 5 minutes. And then that's determined by accumulating enough bursts, single. So that would be something like 6,000 bursts, gives you a nice smooth curve like that. So that's where the rapid mixing device comes in, because with some of these, if we get the protein components together just right, we can get rates of helicase that are too fast to detect. It's all done by the end of 2.5 minutes. The reaction is complete. So we do want to improve the time resolution. So if you have the two things you're mixing, say mix the ATP with the protein components in a microfluidic environment with laminar flow, getting them to actually mix and not just propagate with two parallel streams is kind of tricky. So you have to have a very tortuous path in the device, or squeezing by a very tiny aperture so that you get hydrodynamic squeezing to make the diffusion times very, very narrow. And so that's what that one bioengineering student is making progress on. So then if you mix them in a mixer so that the time scale of the mixing is sub 1 second, and then you have it continuous flow, then you have the confocal detection volume just after the mixing is finished, and then you can move it further and further away to get the time scale. So people have done this. Our mixing devices are down in the sub 1 second time scale, which is much better than a few minutes. And so that's the way we're approaching that.
Are you limited by time there? I mean, because ideally, you kind of like to see this, what I want to call it, subcluster activity, not the entire cluster, but to get this idea that I think we're showing at the end, where you've got multiple ones of these little subclusters attacking the helicase at the same time.
No, it's what I'm saying, within a cluster, there's multiple proteins that are attacking, but all that is happening within each cluster.
So, do you think that's resolvable ever? I suppose is my question.
Each burst gives a lot of information about the dynamics. There's something called burst variance analysis, BVA, where the intensity of each burst has a expected concentration dependence on the duration of its occupancy. And you get this curve in the BVA analysis. If the amount of variance is enough above that, then it indicates that the motions are happening within that 1 millisecond of occupancy time. So that's one way. And there are many other ways of doing it. Another way of getting an intermediate time scale is, suppose the molecule diffuses out of the beam, its likelihood of diffusing back into the beam is pretty high. So you get, you can measure you can estimate the number of repeats, and that is on a smaller time scale than this few minute time scale. So those can get you down into the millisecond time scale. And we do know from the BVA analysis that we have done that the variance of the intensity of each burst is much higher than you'd expect if it's static. So it's clearly millisecond dynamics within the cluster of the fluorophores getting closer and further away.
So the information is there, it's just getting to.
Yeah, there's also a hidden Markov method of doing this, where you can have a model, you have so many reaction steps that you assume, and then you can apply that to the dynamics of each burst. It's a fairly active field, this, of people who love their computers doing different kinds of analysis. And the one we're mainly using, this comes from UCLA, Shimon Weiss and Michaelis, who it's called Fret Bursts. And it's a Python Jupyter pages scripts. And it takes some getting used to, but then it really is very effective. And once you have a script that works well, then the analysis is very quick. So she's very good at this, Shweta, and I did a fair bit of Python programming in that.
I'm really amazed at how much information you get from effectively a single source.
Each burst, yeah, that's right.
Yeah. Any other questions? In that case, let's
Okay,
take care of
thanks.
the talk. Thank you
Okay.
very much.